Thanks for joining us in another fascinating journey through the world of science here on the Sands Channel podcast.
Great to be diving in again.
Yeah, absolutely. And if you're finding these deep dives intriguing, please take a moment to hit that like button and subscribe. Your support really helps us bring you more of this stuff.
It really does.
So today we're about to unravel a bit of a scientific puzzle. It's all about our muscles and how they age. And right at the heart of it is this molecule, NAD. You've probably heard of it. Nicotinamide adenine dinucleotide.
It pops up a lot in health discussions.
It does. Think of it like a tireless little helper inside our cells, especially muscle cells. It's crucial for making energy, the processes that generate ATP.
The cell's fuel.
Exactly. Now, the sort of standard story has been that as we get older, our NAD levels drop in our muscles.
Right, and that decline is often blamed for muscle weakness, sarcopenia.
Yeah, that age-related muscle loss. But what if that isn't the full picture? What if our muscles are maybe tougher than we think?
More resilient.
Precisely. Today, we're diving into some really compelling new research that's, taking a second look at this whole idea. We'll explore a study where scientists did something pretty bold.
They did. Researchers at the University of Copenhagen and some partner institutions, they set up this really fascinating experiment.
Gah.
They used a specific technique, an inducible knockout, to essentially turn off a key gene. It's called Nampt.
Nampt. Oh.
And they did this specifically in the skeletal muscles of adult mice. Now, Nampt is really vital for making NAD.
So switching it off would lower NAD levels.
Dramatically. They managed to get an 85% drop in NAD plus levels just in those muscles.
85%, wow. Okay, that sounds like it would just devastate the muscles. I'd expect them to be incredibly weak, tire out instantly.
That's what you might think, right. But the findings were quite surprising.
How so? Did the mice just, give up moving? Become furry couch potatoes?
Not at all. Despite this huge NAD depletion, the researchers saw basically no significant problems with the muscle structure itself. The morphology looked fine.
Okay, so the muscles look normal, but could they still, work? Could they generate force? Keep going.
No, that's the crucial part. They tested muscle contractility very thoroughly.
Right.
How well the muscles could generate force, both in single twitches and during sustained contractions. And remarkably,
yes,
it was largely unaffected.
You're kidding. With only 15% of the normal NAD, their muscles were still pulling with normal force.
Pretty much, yeah. It really makes you pause and rethink things, doesn't it?
It absolutely does. What about exercise? Could they still run?
They put them through their paces, wheel running, treadmill tests. Again, no significant difference in their exercise tolerance compared to normal mice.
That's incredible.
And get this, in one specific muscle type, the EDL muscle, it actually showed better force recovery after repeated contractions.
Better recovery, so less fatigue
What?
With less NAD. How? How does that even work? That seems completely backwards.
It does seem counterintuitive. The researchers floated a possible explanation linked to glycogen.
The stored glucose in muscle.
Exactly. They found higher levels of glycogen in these NAD-depleted muscles. So maybe the muscles were just, leaning more heavily on that readily available fuel source.
A shift in fuel strategy, maybe. Okay, but NAD is so tied up with mitochondria, right? The powerhouses. Surely an 85% drop overall, even if less in the mitochondria itself, must have hammered them.
Well, they looked very closely at the mitochondria. And even when they measured NAD inside the mitochondria, where it was down by about 50%.
Still a big drop.
Still a big drop, yeah. But they found no impact on mitochondrial structure, and crucially, no impact on their ability to produce energy, the respiratory capacity. It was normal.
Half the NAD in the power plants, and they're just humming along fine.
Seems like it. It seriously challenges that idea that NAD is the absolute gatekeeper or bottleneck for muscle energy production.
Wow, okay. Anything else on the mitochondrial front?
Yeah, they also checked the inner mitochondrial membrane potential. That's vital for making ATP. Unchanged. It suggests the mitochondria are just remarkably adaptable. Or maybe that resting NAD levels are normally way above what's strictly needed for baseline function.
This is really shifting my perspective on muscle metabolism. What about the whole body? Skeletal muscle is a huge player in overall metabolism. Did messing with muscle NAD cause wider issues?
Good question. They looked at the whole body physiology and found, not much difference. Overall body weight, body composition, Muscle to fat ratio and total energy expenditure were all pretty similar to the control mice.
So basically their overall metabolism seemed okay despite this major change in their muscles.
Largely, yes. They could even handle short bursts of intense exercise,
Yeah.
okay. Although there was one little finding, slightly higher blood lactate levels during that really intense exercise.
Maybe a sign of slight metabolic stress under peak load.
Perhaps a subtle shift in how they were generating energy right at their limit.
Interesting. And what about that glycogen again? You said they might be using more of it. Did that affect their ability to refuel afterwards?
It seems they did burn through glycogen faster during contractions compared to normal muscles, but their ability to replenish those stores after exercise, that was completely normal.
So they used it faster, but could stock up just fine afterwards. Muscle adaptability really is the theme here.
It seems to be.
Which brings us to the really big question. Aging. The whole reason NAD gets so much attention is its linked to aging, especially muscle aging. So what happened long term with these low NAD mice? Did their muscles age faster?
This, for me, was maybe the most striking finding of the study.
Yeah.
They look at mice with this lifelong NAD depletion in their muscles.
Right.
And they found zero evidence that it accelerated muscle aging.
Zero. So their muscles didn't get weaker or smaller faster than normal mice as they got old?
Nope. Not functionally looking at size and force production with age.
Yeah.
And not even at the molecular level when they looked at protein profiles and other markers within the muscle.
That really flies in the face of the common narrative, doesn't it?
It absolutely does. In fact, the study concluded that age itself was a much, much stronger driver of changes in the muscle than the NAD level was.
So getting older changed the muscle way more than having low NAD did.
Precisely.
Okay, one more angle. What about reactive oxygen species, ROS? Those damaging molecules sometimes linked to aging and exercise stress. Did NAD levels affect those?
Well, at rest, ROS production was similar. But after exercise, the NAD-depleted muscles didn't show the expected increase in one specific type of ROS lipid hydroperoxides.
So maybe NAD does play some role, perhaps in how muscles adapt or respond to the stress of exercise, even if not critical for baseline function or aging rate.
That could be. It suggests a more nuanced role, maybe not the central one we assume for basic function and aging, but perhaps involved in specific signaling or adaptation pathways.
Were there any other sort of molecular clues? Did other related molecules change?
They did note that some NAD breakdown products like ADPR were lower. This suggests that maybe some enzymes that use NAD were less active, which makes sense if there's less NAD around. But another key process often linked to NAD, called global protein acetylation, that seemed unchanged.
Okay, so putting this all together, after this deep dive into this really quite surprising study, what's the main takeaway message for us?
I think the biggest takeaway is that skeletal muscle seems way more resilient to NAD depletion than we thought. Much more.
Okay.
This study showing pretty normal muscle function, mitochondrial health, and crucially, no accelerated aging, despite a massive NAD reduction. It strongly suggests that the simple idea of age-related NAD decline being the main cause of muscle aging is, well, probably too simple.
So other factors are likely more important.
It certainly looks that way. Other metabolic pathways, maybe changes in repair mechanisms, inflammation. The picture is complex.
This really makes you question the whole strategy of just trying to boost NAD levels in muscle to fight aging, doesn't it? Like, it might not be the magic bullet some hope for.
Exactly. It suggests that approach might be less impactful than we thought. Or at least it might need to be combined with other strategies. It definitely opens up new questions and avenues for understanding muscle health and aging.
It certainly does. It's a reminder that biology is rarely straightforward. This study gives us a lot to chew on. Thanks for guiding us through it.
My pleasure. Science is always evolving. It keeps things interesting.
Absolutely. As always, don't forget to like the video and subscribe if you haven't joined us yet.